It will be a good reaction. Yes. You look too hard.
Right, I think I'm live. Can everybody hear me in the back? Great, fantastic. Excellent. Well, ladies and gentlemen, my name is Maurice Gleeson, and today I'm going to talk to you about the autosomal DNA, which is the family finder test was just mentioned. It's used in adoptees and finding long-lost Irish relatives. So, to start off with, what will we cover? Well, first of all, I want to talk about what is our goal. That is a very, very important question to ask when you're talking about any kind of DNA test. What is the goal of this test? What am I trying to achieve? The second one is what is autosomal DNA, or AT-DNA as it's frequently called. I'm going to talk about the two approaches to using autosomal DNA, either to address a specific question or just to go on a fishing trip. I'm going to talk about what is a match. I'm going to talk about a step-by-step approach to analysing your matches. I'll also be mentioning DNA adoption, which is a group in the US that is having great success using autosomal DNA to put adoptees in touch with their biological families. And then I'll also mention about the future directions for autosomal DNA, because it is still quite a young test. So the first question, what is our goal? And the goal with autosomal DNA is quite simple: to connect with distant cousins and to use what they know about their family tree to break through brick walls in ours. So in a nutshell, that's what we're trying to do. Now, here is my family tree. And these are the brick walls that I have in my one. You can see that I've identified about half of my great-great-great-grandparents, but there are still large gaps in where I've done. And that's after I've exhausted the family lore, the letters, the Bible, the 1901-1911 censuses, Griffith's evaluation and all the cancelled books in the evaluation office, civil registration records, church records, collaboration with other ancestors, online newspapers, Glasnevin records, the 1939 electoral roll. These are all the records that I've currently used, and I've gone as far as I can with the documentary evidence. And I have hit a brick wall. And this is where a lot of people doing Irish research will stop, because they hit that brick wall and you can't get through it. Now, in my particular case, I've used Y-DNA on my Spearin branch to break through that brick wall and take me all the way back to the 1660s. Now, it's still a little bit patchy, but it's pointed me in the right direction, and it tells me you need to start searching in this area. You need to focus your research, and it'll take you back a little bit further. Now, with an adoptee's brick wall, this is the sort of picture that you'd find. Many adoptees, they might know who their birth mother is, but they don't have no idea who their birth father is. Are there any adoptees in the audience? No adoptees in the audience?
We're working on a case.
Are you?
Exactly like that.
Well, this is a very typical case. Certainly there's two cases in my family tree where someone was born out of wedlock, and we found the birth certificate with the mother's name and then just a dash for the father. In some cases, you'll find that adoptees don't even know who their mother is. So this is the kind of situation that an adoptee is facing. An adoptee's brick wall is immediately after themselves, whereas our brick wall normally stops about 1800. But the techniques that they're developing in the US for looking at adoptees and helping them break through the brick walls could also help us in our Irish research breaking through the brick wall of the 1800s. Now, there are a lot of things that adoptees in Ireland can actually do. There's the contact preference register, where you can say, I do want to be contacted by my birth family if they want to contact me. And similarly, if you know of people in your family tree who have been adopted, you can actually, as a natural relative, put your information up on the register and say, I do want to be contacted by these long-lost relatives if they want to get in touch with their birth families. Bernardo's run a post-adoption service here in Ireland, but what happens if the adoption was prior to records in 1954? And my dad tells a lovely story about how his mum was asked by a friend to investigate an ad in the Irish Times, baby available for caring family. And that was back in the 1910s, 1920s. and she went along to the family, they had a little chat, and they left with the baby. So the things have changed since then, but what hope would that person have of finding their family? DNA is an additional tool to help the adoptee find their to find their birth families, but it doesn't guarantee an answer. It can help point you in the right direction, and like I say, the adoption community in the US have been having a lot of success, but is it possible in Ireland? Or is the 1800 brick wall going to stop us from actually getting any further? There are some very good books that I would certainly recommend. This one by Richard Hill called Finding Family is a riveting read, and he combines documentary research with DNA evidence to actually find his birth family. So if you're interested in this topic, certainly get this book by Richard Hill. If you do the autosomal DNA test yourself, even if you've got nothing to do with adoption, you will be contacted by adoptees. And here is an extract from an email that I received from a lady. Unlike people who are doing their tree out of interest, we have a specific purpose, which is to find out who my mother's birth parents are. Only this year, we received a letter my grandmother had written in 1962, which a relative in Australia had kept all these years. She also sent photographs of my mother's mother, so at the age of 84, she finally got to see what her mother looked like. You never know what you will find. And this is the lady. Pauline May Cartwright is the 84-year-old woman. Eric is her brother. And her mother is Phyllis Ina Cartwright, born 1904, died in 1970. And at the age of 84, this is the photograph that she received of her mother. So you never know what you will find. And that is going to be a key feature of autosomal DNA testing. You will unearth cousins and distant relatives that you never knew existed. So what is autosomal DNA? Well, there are three types of DNA tests, as you've heard over these last 2 days. The Y DNA test takes us back on our direct male line, our father's father's father. And then on the opposite side of the family tree is the mitochondrial DNA test, which is the direct female line, continuous female line, mother, mother, mother. And right in the middle, we have the autosomal DNA, which takes us back about 7 generations only, so it's a much more confined reach. This is my family tree. I've done the autosomal DNA test, so it's going to take me back about 7 generations to about 1765, a common ancestor born in 1765. My dad has done it as well, and so has my maternal aunt. So between the three of us, we actually managed to get back to about 1730 or so. So if we match somebody on the autosomal DNA, it could potentially put us back in touch with a common ancestor born about 1730. Now, let's take a little more in-depth look at the DNA. When you do your sample and they send it off to the lab, they extract the cells from that sample, and in those cells you have the mitochondria, which give you your mitochondrial DNA, but in the nucleus of the cell you have the 23 chromosome pairs, 46 chromosomes altogether, and the last pair, chromosome 23 pair, is the X chromosome and the Y chromosome. These are known as the sex chromosomes, because women will have 2 X's and men will have an X and a Y. And here's another image of the chromosomes. The chromosomes altogether are in 23 pairs. The sex chromosomes, there's just 1 pair of them. The autosomes is everything else that's not a sex chromosome. And there's 22 pairs of autosomes, and that's why it's called autosomal DNA. And those are the autosomal chromosomes there. Now, they account for about 95% of the DNA that's present in the chromosomes. The mitochondria, on the other hand, are these little ones down here, and there's about 1,000 mitochondria in every cell. So you only have 46 chromosomes, but you've got thousands of mitochondria, which is why it's very good for ancient DNA, because you tend to find one of these thousands of mitochondria in the ancient DNA much more likely than you would do the chromosomal DNA. So those are the chromosomes. the X chromosome tends to be included in the autosomal test as well, so you get the X chromosome as well as the autosomes. Now, how much DNA do you actually get from each of your ancestors? Well, here's an example of the 4 times great-grandmother. So say my 4 times great-grandmother, who was born about 1765, she would pass on 50% of her DNA to each of her 12 children. For example, she had 12 children. And so the first child will get 50%, but then he would pass only 25% of great-great-great-great-grandmother's DNA on, and then it gets diluted further to 12.5, 6.25. So by the time it reaches me, I only have 1.5% of my great-great-great-grandmother's DNA. Now, another question that people ask is, how much DNA do I share with my relatives? Well, siblings would share 50% of their DNA in common. First cousins only 12.5%, second cousins 3%, third cousins 0.8%, and by the time it gets down to me, fifth cousins only share about 0.05% of their DNA. That seems very small, but DNA has got millions of bases, so you're still looking about 30, that is actually millions of base pairs, that 0.05% there. Now, the other question is, how many cousins will the test detect? And here we can see that it will detect about 99% of first and second cousins, about 90% of third cousins, and 50% of fourth cousins, but only 10% of fifth cousins. So the further back it goes, the less likely you are to detect somebody. But because we actually have more fifth cousins than we'll ever have first cousins, and if you assume that every family has 3 children in the family, and those children have 3 children, and those 3 children have 3 children, by the time you get down to fifth cousins, you've got far more fifth cousins than you would have second, third, or fourth cousins. And that's why when you do the test, you'll actually find that most of your matches are fifth cousins and fourth cousins, primarily fifth cousins. So that just gives you an idea of what to expect when you get the test. You won't have that many first, second, and third cousins. It will be much more the distant cousins. But in terms of your success of actually hooking up with a common ancestor, you're going to have much more success the closer that relationship is to you. Now, I talked about the mystery of the wedding memento yesterday. This was a memento that my dad found in his grandfather's papers about a marriage of David Patterson to Ruby Kathleen Gleeson. They were married in Thargomindah on the 23rd of March, 1893. And I looked at Thargomindah on the map, and it is right in the middle of the Australian desert, surrounded by man-eating koalas, mosquitoes, scorching desert, and teetering on the brink of nothing. So I was totally flabbergasted. What was a wedding memento from a marriage that took place in the Australian desert doing in my great-grandfather's papers in Dublin? So I set myself, or rather my dad set myself the task of trying to trace who this person was. So I went in search of Ruby Kathleen, and I found her death certificate and her marriage certificate, and showed that her parents were John and Anne Gleeson. And I thought, fantastic, because my great-great-grandparents were John and Anne Gleeson. And there were other little bits of clues. She came from Longstone. Oh, that's fantastic. All of my crowd came from Longstone as well, or the neighbouring townlands. And we gradually put together little bits of additional information that suggested that there really was a link between the Ruby Kathleen in Australia and my own Gleeson ancestors in Tipperary. One of whom was supposed to have gone to Australia in 1877 and who had a daughter who was a concert pianist. I traced the descendant of Ruby Kathleen, her grandson, who was in his 80s. And we struck up an email conversation, and he gave me some information about his Australian family history and how Ruby had an uncle and an aunt. There was a brother who was a baseball player, and she also had a daughter who was an accomplished musician and excelled at violin. So there were a lot of parallels between the two stories, but we couldn't tell for sure whether the two were definitely related until we did the DNA. So here's a picture of my great-grandfather, Martin M. Gleeson, and there's a picture of Ruby Kathleen. And if they were siblings, which is what the documentary evidence was suggesting, they would have shared 50% of their DNA, and when it gets down to my dad, he would have shared about 3% of his DNA with Ruby's grandson. And when we did the test, the results came back that they actually shared 2.6% of their DNA in common. So it was able to confirm what we suspected, and that is one of the great advantages of autosomal DNA. You can confirm suspected relationships. I tested as well, and I came back at 1.5% of my DNA being shared in common with Ruby's grandson, well within the range of what you'd expect for a second cousin once removed. So the other interesting thing was I tested my siblings, and there was quite a degree of variation. If you look at my second brother over here, he only had 0.6%, which was 3 times less than what I shared. And that just goes to emphasize the kind of genetic lottery that we are all subjected to. There's no guarantee that we are going to have a particular segment of DNA passing down through the generations to the current generation. And in actual fact, when I looked at this on the family tree DNA page, it estimated the relationship between my brother and Alan was 5th cousin or greater, rather than 2nd cousin once removed. So the estimates that you get on family tree DNA are just that, they're estimates. And there's a large margin, a large range around that estimate that you need to be aware of. Now, what has autosomal DNA testing told us in this particular situation? Well, Dad and Alan are definitely genetically related, and the best fit is second cousins. It's confirmed a suspected relationship with 99% probability. But many unanswered questions remain, but DNA helps focus our research. One of those sisters of my great-grandfather went off to Australia and got married and changed her name to Ruby Kathleen. But who was it? Was it Winifred? Was it Catherine? Kathleen? Okay, maybe. So we need to do more documentary research to still continue with the research that the DNA evidence has helped us unearth. But certainly the DNA testing has helped us save Ruby Kathleen from the shoebox of obscurity. That's the shoebox that you put all of those files in that they're nothing to do with our family, it's a false alarm, under the bed. So she's been saved from that. We have cousins in Australia, they have cousins in Ireland, and it has opened up a whole new area of research for this particular line of the family tree. So that's an example of how autosomal DNA can be used to address a specific question. I'm sure we're related to that crowd and they have money. Somebody better take the test. To answer a specific question, you and someone else have to take the test. If you're just going on a fishing trip, and that's what the rest of my presentation is going to talk about, you just take do the test and see what you catch. There might be relatives swimming around in the database, and you might be able to hook them. So let's talk now about how you actually go on a fishing trip. And there are 3 companies that do autosomal DNA. Family Tree DNA is downstairs. Now, on the surface of things, they all charge the same price, but I'll tell you in a while that there are additional costs associated with 23andMe and Ancestry DNA. But whichever one you test with, you should also join the free website GEDmatch, okay? Now, GEDmatch is run by volunteers. It's no charge to you at all. You can upload your raw data, and it tells you a variety of different things. So you can upload family tree DNA data, Ancestry DNA data, 23andMe DNA data, and it will allow you to analyze the data using a variety of different tools and utilities that they have on the website. You can compare your DNA to choose one other person. You pick one other person, you compare your DNA to them. You can compare to many different people. You can do ethnic admixture. You can look at the X chromosome, very, very important. you can use phasing as well. If you test your parent, you can actually phase your chromosomal data. So do join GEDmatch. These are my results from family tree DNA. And just to orientate you, these are the individual people I match. There's a Hare, there's a Haddon, there's a Spearin, there's a Swords, there's a Maguire. This tells you the date when they joined. This tells you the relationship range that the computer is estimating. And you can see here that Mr. Haddon is somewhere between my second and fourth cousin. It's an estimate, a second or fourth cousin. Could be second, could be third, could be fourth. It could be even more distant than that. You can assign known relationships, and this is my second cousin once removed, so I've assigned that relationship to him. You can see the amount of shared DNA that you share: 104 centimorgans, 61 centimorgans, 66, 45, etc. And then here, this place for people to put in their ancestral surnames. Now, not everybody has done that, and that doesn't really help anybody. So if you are going to do the test, make sure you put in your ancestral surnames. Certainly down here, these 3 people have put in their ancestral surnames, and that gives you an idea. You look at the ancestral surnames that they have, and you see if you recognize any of your ancestral names in that list of surnames. Now, you can also click on the Advanced button, and that will allow you to triangulate, which is going to be very important. We're going to talk about that. It will also tell you what tests they've taken. This chap has taken the Y-DNA 67. This chap has taken the Y-DNA 12 and the full mitochondrial sequence. And you can also compare them in the chromosome browser, so you can compare different people's chromosome segments with each other and see if any of those segments overlap. That is going to be very, very important as well. And then it also tells you the longest block of DNA they have. This one is 34 centimorgans. This one here is 24 centimorgans. So there's a lot of information that you get from your matches page. Now, here's an example of a father-son relationship sharing 50% of their DNA, 2 brothers sharing 50% as well, aunt and nephew 25%, and a second cousin once removed sharing only small segments of DNA. Okay, so the further the relationship, the more distant the relationship, the less DNA that is shared. But I just want to talk about why there is. These are the first 22 chromosomes. Family tree DNA doesn't have the X browser yet, but that is supposed to be coming. Why don't they have 44 chromosomes here then? You know, this is chromosome 22, 21, 23, and I'm going to talk to you about why that is so. And there's a technical reason for this, so I hope you can follow it. Here's your average chromosome. Let's say it's chromosome 21. If we unravel it, we see the classical double helix, and that comes out into 2 strands. And along the strands, we have all these base pairs here, 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 all the way along. Each of them are paired with each other. And these nucleotide bases follow a very, very strict pairing. G only ever binds with C, and vice versa. A only ever binds with T, and vice versa. Guanine, cytosine, adenine, thymine. So because they only ever bind with each other, when we're reading it, we just, because this is a mirror image version of this, we don't need to read this when we're actually reading the test. Now, if we compare our 2 copies of chromosome 21 with our matches 2 copies of chromosome 21, this is how it happens. For each marker each position that we're looking at, this position would be T on top, T below, so it's TT. Remember, you've got 1 chromosome from your father, 1 chromosome from your mother. This position here, we have 2 Gs, G and G. This position here, we have an A and a G. This position here, we have a C and a C. On the matches chromosomes, you go through the same process, and maybe their maternal one is T, their paternal one is T, so it's a TT. Their maternal is C, is T, the paternal is C, so you get a CT. Here you've got a G and an A, maternal, paternal, and here a G, a C and an A, maternal, paternal. So when we come to compare these two, what you find is that the T and the T are known as a fully identical region. The machine that's reading these is not able to distinguish between maternal and paternal. So it says that yes, you've got a T and you've got a T in both the U and the match. For the next one, I have the GG, my match has the CT. They don't match at all. So the machine says no match. It's non-identical. For the next one, I have AG, my match has GA, but the machine doesn't worry about the sequence. So it says yes, this is a fully identical region. And for the last one, CC and CA, the machine says yes, there's a C there, but there isn't an A. So we only got a half match there, and that's known as a half identical region. And I'm using those colors deliberately because if you look on GEDmatch where you match, this is the sort of graph that you will get, and you'll have the non-identical regions in red, the fully identical regions in green, and the half identical regions in yellow. And it's those regions that are either yellow or green that the test will detect as an identical DNA segment. Okay? So for each chromosome, we're comparing your 2 copies of that particular chromosome with your match's 2 copies, maternal and paternal, of that particular chromosome. And the half-identical regions indicate that one of your copies and one of your match's copies have identical bases at that particular segment. But we don't know whether it's on the maternal side or the paternal side, the maternal chromosome or the paternal chromosome. But the longer the segment, the more robust the match is. If we look at the major companies that are providing these type of tests, autosomal DNA tests, the GEDmatch, in terms of those that allow uploading of raw data, it doesn't apply to GEDmatch, but Family Tree DNA, 23andMe, and Ancestry will all allow data download. So you can download your data and then upload it to GEDmatch. But the problem with raw data upload, Family Tree DNA will also allow you to upload both 23andMe and Ancestry data into their database. So that allows you to fish in 2 different pools. But Family Tree DNA are the only company that allow you to do that. Chromosome Browser is present. They all have a chromosome browser except Ancestry. It does not have a chromosome browser, so you will not be able to compare your DNA on Ancestry. X chromosome browser is on GEDmatch and on 23andMe. Family Tree DNA are getting one soon. Ancestry do not have one. And in terms of cost, all of the tests cost $99, but with extras like postage and packaging, it brings it up to $115 for Family Tree DNA, $250 for 23andMe, because you have to pay about $80 to get it posted to you. And then if you want to upload it to Family Tree DNA, it's an extra $70 as well. And then for Ancestry DNA, it's only available in the US. You can't get it in Ireland, so you'll have to jump on a plane. I priced the cheapest I could find, and I'm assuming that you land in JFK, somebody hands it to you, and you jump back up on the plane afterwards. So that'll bring it up to about $630 or so. Now, let's talk about what is a match. What makes a match a match? In short, a match is someone who has an identical segment of DNA compared to you. And that implies that the 2 of you share a common ancestor at some point in the past. And that common ancestor has passed on this particular identical segment to both of you over the course of time. It's wound its way down through your ancestry, and you and your match just happen to be lucky enough to have exactly the same segment. And that segment is called an identical by descent segment, because it has descended to you, and that same segment is identical in you and your match. When is a match not a match? When it's a false positive. And these happen a lot of the time. It's more common as the segments get shorter. As the segments get shorter, the number of false positives increase. So, and there are different kinds of false positive. It could be just identical by state. There's a particular false positive called a fuzzy boundary, where you have a true positive, yes, it did descend from a common ancestor, but it has a false positive and added to the end. So it looks longer than it should be. Then there's something called compound segments, where you have something on your paternal chromosome, something on your maternal chromosome, and just by chance, the two match up with somebody else's DNA, giving you a false impression that it is the same segment, when in fact it is not. If you want to read more about it, see Anne Turner's article, and also the Family Tree DNA Frequently Asked Question number 612. I've put it up there at the bottom of the slide. This is being recorded, by the way. This presentation will be available on YouTube, on the Genealogy Ireland 2013 YouTube channel. So you might want to watch this again, and some of you might want to watch it again and again. So, this is a slide from Tim Jansen, and it really just emphasizes the fact that the shorter the length of the matching segment, the greater the chances of it being a false positive, right? So if I just point out a few things to you, if you get a matching segment that is 8 centimorgans large, you only have a 50-50 chance of it being a true positive. You've got a 50% chance of it being a false positive. So be very wary of any matching segment below 8 centimorgans in size. And this is when you take it in isolation. If there's documentary evidence that the two of you are probably related, then that makes it less likely that it'll be a false positive, but you still have to be very, very careful with these. If we get up to 10 centimorgans, then we find that the chances that a segment is identical by descent, and therefore a true positive, goes up to 99%. So I would encourage anybody, when you're first looking at your matches, try and go for the largest segments and focus on segments that are greater than 10 centimorgans in size. At the far extreme, if you go down to 4 centimorgans, you've got a 99% chance of it being a false positive. So try and avoid the smaller segments when you're talking with your matches. I frequently get people saying, oh, we match in our DNA. We've got a 3 centimorgan segment in common. And I'm going, well, that's fascinating, but I don't think it's enough to confirm a relationship. So that is the problem with smaller segments. Now, it's important when you're dealing with your matches, I think, to take a step-by-step approach. And this is an approach that I've developed myself. And it's really just to try and make things easier or as simple as possible. The good news is you don't have to follow all of these steps, number one. Number two, you can follow the ones you want to and discard the ones you don't want to. And number three, you can get as technical as you want to. It depends very much on what your own particular preferences are. But the steps in my approach are, number one, where does the common ancestor sit? Number two, is the common ancestor obvious? Number three, eliminate non-contenders. And number four, work with small related groups. And that means identifying in common with matches and their overlapping segments, which I'll talk to you about, comparing trees, and then creating a network of related collaborators, and eventually harvesting trees and GEDCOMs or GEDCOMs, and using the GEDCOM comparison utility developed by the DNA adoption group in the US. And then repeating steps 1 to 3 with each of your matches, and not just with one of them. So it can get very involved, but I'm going to run through each of these steps, and you can decide what level of involvement you want. And like I say, you can watch this again and again and again on YouTube until you're blue in the face. And I can guarantee that many of you will be, if you're not already. So let's start off by looking at the different kind of tactics that we're going to use. Some of these tactics involve DNA, some of them involve genealogy, and some of them involve sitting around a card table and collaborating with a group of people you match to see if you can find a common ancestor. And a lot of the time, working with these groups is like playing a game of poker. You've got your cards, your deck of ancestors in front of you, and you discard the ones that you're eliminating as non-contenders, And then finally you show your hand and you see if anybody else around the card table goes, oh, I've got a queen too. That really is the approach. The secrets to success with autosomal DNA testing are to start with your closest matches, not your furthest matches, your closest matches. You have to share your tree. It's not good enough sharing surnames. You have to share a tree that goes up to the furthest you can go and that comes back down again. It's the branches that come back down are very, very important. You need to develop a relationship with your matches. It's no good just pinging off an email saying, oh, the DNA says we're related, what do you think? You do have to develop a relationship with them that evolves over weeks and months. And you need to keep track of what you've done, because it is very easy to lose track with the 1005th cousins that you're going to get hold of. So the first step, where does the common ancestor sit? And this is all about just positioning your common ancestor on your family tree. And this is nothing to do with DNA, this is purely genealogy. So if you don't know much about DNA, this is an easy step to start off with. And here's an example of me in 1960 and my match who was born some other time, and the test says we're third cousins and we share a great-great-grandparent. And that would mean that we've got one of our two times great-grandparents born around about 1835. We share that ancestor in common. Of course, that ancestor is married to Mr. Ancestor. So even though we have a lot of ancestors at that level, we've only got half the number of ancestral couples at that level. And that's quite handy. It does narrow the odds a little bit as I go. Now, I have 16, we all have 16 great-great-grandparents, but they're divided into 8 couples. If we were just looking at a single person, we'd have 16 by 16 possible combinations for the common ancestor, in the sense that, okay, I've got 16 possible common ancestors, you've got 16 possible common ancestors, 16 by 16 gives 256 possible combinations. My ancestor 1 could be the same as your ancestor 1, or your ancestor 2, all the way up to 16, and I could do the same for all 16 of mine as well. But because they're in couples, we only have an 8 by 8 possible number of combinations for the ancestral couple. If we go up to 5th cousins, then the situation gets even more complex, and we have 4,000 possible combinations of linking up common ancestors, but only 1,000 possible combinations for the common ancestral couple. So this is important to find out exactly where your common ancestor sits, because that gives you an idea of the range of possible combinations that you will have with your match. The other point to make is that your match might be actually a lot further out than you think, because the test will suggest a certain relationship, 1st, 2nd, 3rd, 4th, 5th, and the actuality may be a lot different. Certainly with 1st, and this is just made up figures, but it serves to illustrate the point. When you get up to, say, 2nd cousins, it will be 90% accurate, that 90% of the suggested 2nd cousins will turn out to be 2nd cousins in actuality. But it gets less as time goes on, and there's a greater spread between the actual relationships as you get further and further away. So with 5th cousins, you might only have 30% of your actual 5th cousins would be suggested as 5th cousins. You might get 20% of them would actually be 6th cousins, and 20% would be 7th cousins. So the important point here is that the suggested relationships become more inaccurate the more distant they are, and certainly with 5th cousins, you're more likely that if the test says it suggests that you are 5th cousins, you're more likely to be 6th cousins or 7th cousins in actuality. And the common ancestor is likely to be further back than you think. The greater the distance in between you and your match. We also have to, there are other factors that we have to keep in mind that would make the connection further back than it actually seems, such as cousins intermarrying, and that would never happen in Ireland. if that actually does happen, then more DNA persists than is usual. So because there's more DNA between you and your match, because you both have cousins intermarrying, it makes you look much closer related than you actually are. And that's an example of pedigree collapse. The further you go back, you find that on paper you should have loads and thousands of ancestors. In actual fact, it's a lot less than you think, because cousins will inadvertently intermarry as you go back in time. Then we have the problem of endogamous populations like the Ashkenazi Jews, the Romani people, Hindu castes, and Jehovah's Witnesses, who tend to marry within the community. So anybody who has Jewish heritage will find that they have thousands, literally thousands of matches. I have 250 matches on autosomal DNA. My dad has 250 matches. My aunt has 250 matches. It's about the Irish average, I would say. But a Jewish person will have 1,000, 2,000, 3,000 matches because of this endogamy, this intermarrying between cousins and staying within the same close-knit community. The next step in this process is the common ancestor obvious? And the only way to do that is to make contact with your match and to compare trees. And, oh, yeah, there he is. I see the common ancestor. Can you see him? Yeah, there he is up there. That's what you're looking to do at step 2. You have to develop this relationship, you have to contact your match, and you have to compare trees. And when you're comparing family trees to find the common ancestor, if both of you have extensive genealogies, you can go all the way back up and you just look through your ancestors, you think, oh right, there you go, you said Roger Doyle and Annabel Tubridy, yeah, I've got them on my tree, that's where we must be related to. Unfortunately, that is unlikely to be the situation in Ireland, because many people cannot identify Irish ancestors beyond about 1850 or so, and we have a brick wall. And so we've got a brick wall on our side of the family tree. So what does that mean if somebody else has an extensive genealogy, but we have a brick wall right there? Well, we use the example of Roger Doyle and Annabel Tubridy. Let's say that Doyle and Tubridy had a daughter. The circles are female, the squares are male. And her maiden name was Doyle, but she married a Ryan. And they had a daughter whose maiden name was Ryan, who married a Spearin. And they had a daughter whose maiden name was Spearin. Now, I'm lucky in this example because I actually know the maiden name of that distant ancestor. But for a lot of my great-grandmothers, I don't actually know what the maiden name was. It's great-great-grandmothers. Once you get, and that's, I think, common with a lot of people, you get back to your last ancestral couple before the brick wall, you know the man's name, you know his surname, you know the mother's first name, but you don't know her maiden name. So what happens in this kind of situation? Well, in this kind of situation, I would not have any connection between my grandmother Spearin and the most distant known ancestor in my match's tree, because it's Doyle and Tubridy. I'm trying to connect Spearin with Doyle and Tubridy, but my brick wall is stopping me from actually getting there. Now, even if the brick wall was one generation higher, and it was Ryan, I'm looking for a Ryan and trying to connect it with a Doyle and a Tubridy, still no luck. The only way, and this is why it's so important to trace down as well as up, is if my match has been very good about tracing their family tree and has actually traced from their most distant known ancestor down as well, and has identified that one of the Doyle daughters married a Ryan, then I could possibly connect my Spearin Ryan most distant known ancestors with the Ryan Doyle ancestral line in my match. So it's really pot luck. If you're just going back up on a pedigree chart and you're just stopping at the most distant known ancestor and you're not coming down, then that's going to create problems. If, in this particular situation, well, you have to share trees and not surnames, so it's also a very good idea to have your tree available on either Ancestry or RootsWeb, and to make it available to the people that contact you to explore your match. Now, in another situation, for example, and this happens in Ireland a lot, we go back to about 1850, you get two people who are bricked wall around about the 1850 area. What can you actually do in this situation? Let's take another example then. Supposing the common ancestor is a Spearin marrying a Morgan, and there's a direct male line down here, so the son, the grandson, the great-grandson have passed the Spearin line down along past the brick wall, so it's penetrated the brick wall. If, for example, and I'm just going to show you here, on this side there are daughters, so the name has not penetrated, okay? I'm unlucky in this situation because there's no possible way of connecting back up to this common ancestor. But if it was a male line and one of the daughters was called Spearin, this would be an example of how the name penetrates the brick wall on both sides of the line, both me and my match's line, and in that sort of situation, we are lucky enough to be able to say, oh, the connection is probably a Spearin connection, because you've got Spearin in your tree, I've got Spearin in mine, but it's relying on the fact that there's a direct male line descent that penetrates the brick wall, and that doesn't happen in a lot of families. So step 3 then is to eliminate non-contenders. And there are several steps here. You can eliminate maternal, paternal. You can narrow down to a specific common ancestral couple. You can look at the match on the Y DNA, the mitochondrial DNA, a match on the X chromosome, very important. You can look to ethnicity, nationality, and geography to help you rule out non-contenders. And you can also phase your grandparents to rule out non-contenders. So let's have a look at some of these steps. And again, we're going to use the tactics of DNA and genealogy. The goal is to get rid of your ancestors, to discard any ancestors who could not possibly be the common ancestor with your match. You're going to whittle down the number of potential candidates. Each ancestor in this particular photograph is identified by anantafel numbers, which is the standard way of numbering your ancestors. I'm 1, my dad is 2, my mother is 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. It goes all the way back there. Men are even, women are odd. Sorry, sorry about that. Had to get that in there. Fathers are double their sons. So if you want to calculate the number for a father of an individual, you multiply it by 2. If you want to calculate the mother of an individual, multiply it by 2 and add 1. So if you can, using the technique of maternal versus paternal, you can discard one side of your family. Great, you just got rid of half your ancestors. Brilliant. Using the technique of a match on the X, you can discard another couple of cards. If you look at ethnicity, geography, nationality, that can maybe whittle it down a little bit further. And when you phase your grandparents, that can narrow it down even further. You can discard 25% of your ancestors. Now, one good way of deciding maternal and paternal, and we've done it in our family tree, is myself and my dad have done the test. Now, if a person matches me and my dad, well then the common ancestor has to lie on my paternal side. That's pretty obvious. But if a person matches me, but not my dad, then the common ancestor has to lie on my mother's side. So that's a very, very good way of ruling out half of your ancestors with just by testing 2 people. Triangulation, you can take it a step further, and these are all of the people in my family that I have actually tested. So I've tested 9 people in the family. Here I am here. There's my dad, his sister. There's my mother's sister on that side. Second cousin once removed, second cousin, that'll be a second cousin once removed, that's a second cousin, and a third cousin over here. So there's a lot of people in the family who have been tested. Now, if I have a match between, if my dad has a match with somebody, and also his second cousin matches the same person, then effectively what that means is, and if we know that dad and his cousin match each other through their common great grandparents, John and Anne Gleeson. And by doing that, we're able to eliminate a sizable portion of our ancestors from consideration. So, and we know that the common ancestor between all three of those matches must lie somewhere above John Gleeson and Anne Gleeson, my dad and his cousin's great-grandparents. So testing multiple family members enables triangulation in common with matches. Dad shares a match in common with his cousin. So it's a three-way match. A matches B, matches A. The A matches B, matches C, matches A. And if these are present, then it's highly likely that the common ancestor to all three people sits at or above the common ancestor of you and your cousin. It's useful just to recap on the pathways of DNA transmission. And this is the Y path coming down here. Very useful for adoptees. If you don't know who your father is, have your Y DNA tested, and people who match you are like somewhere in that group of matches is likely to be the name of your birth father. And that's exactly the technique that Richard Hill did and writes about in his book. On the other side, you have the mitochondrial DNA, and this is my mitochondrial DNA here, on this side here, and this is my dad's mitochondrial DNA here. Now, it's interesting that the Y line, myself and my dad have exactly the same Y DNA, but my mitochondrial DNA is completely different from my dad's mitochondrial DNA. And of course, the mitochondrial DNA is passed on to the boys as well, but they don't pass it on to their children. Mitochondrial DNA is just passed on by the women. And why is that? Well, if we go back to the cell, and I'll just bring that up here, it has the 46 chromosomes, as you can see, concentrated in the nucleus, and these little blue things here are the mitochondria. And in women, when they make an egg cell, the cell divides into two, splits into two. Half the chromosomes go down to the nucleus in the egg, but also the mitochondria pass down to the egg. The cell is just split in two. Half the chromosomes go to the new cell, and half the mitochondria go to the new cell. Now, it's quite a different situation in the male, because when a man produces a sperm cell, all of the chromosomes are, again, it splits into two sperm cells, and the 23 chromosomes go down just to the head. But you can see how small this cell is. It's really just a head and a tail. There's really not much to it at all, compared to this rather large, chunky cell that you see down here. And when the sperm cell fuses with the egg cell, it passes its 23 chromosomes back into the egg, and between the two of them, the 23 chromosomes in the egg and the 23 chromosomes in the sperm cell unite to give you your full complement of 46 chromosomes altogether. But that explains why the mitochondria is useful for tracing your female line, because you only get your mitochondria from your mother. The X-DNA is also a very useful way of looking at, is very, very helpful, because I'm an X and a Y, so that's me down here. I will have got my Y chromosome from my father, so there he is there, and my X chromosome from my mother. Now, my mother will be two X chromosomes, one from her mother and one from her father. And if we look at her mother's line, she got one from her mother and one from her father, and it goes back up that way. And if we look at my mother's father, he will only have got it from his mother. So you see what's happening. As soon as we hit a male, the path of transmission, the path of inheritance stops, and it only continues up the female side. Same here, we hit a male, it only continues up the female side. And this makes it very, very useful for ruling out non-contenders, because if you match somebody on the X chromosome, then that means that there are only certain ancestral pathways down which that X chromosome could have passed. And and in terms of the number of ancestors that it could possibly be, you only have 50%, the generation immediately above you. But if you go back to your 5 times great-grandparents, only 16% of your 5 times great-grandparent would be contender to be the actual common ancestor with your match. So if you and your match have a match on the X chromosome and share a segment of your X DNA, that means that it has to be from only 16% of your 5 times great-grandparents, and the other 84% you can rule out immediately. So it's a very, very useful technique to do. Now, I've mapped it out, right? This is me, mother, father, grandparents. So it's a fan chart. It goes all the way back. And within the red area here are the ancestors through which the X could have passed. All of these ancestors that are not within the red area, there's no way at all that I could have got my X chromosome from them. So if I have a match on the X chromosome, I can rule out all of these people as contenders. I've done the same thing for my aunt, and this is hers here. The female is slightly different because she inherits one of her X's on her father's side and one of her X's on her mother's side. But for a male, and I'll just go back to that, for a male, it means that a match on the X rules out your entire paternal side of your family. So it's really, really helpful. So that's the X chromosome. And as far as autosomal DNA is concerned, we've talked about this before, this is the amount of DNA you will have received from any specific of your ancestors. There's 2 things that we need to talk about with DNA and autosomal DNA. First is independent assortment, and that means that people will, when the chromosomes line up before they're split apart, they line up in their pairs, and then one chromosome from each pair goes into one cell, the other chromosome goes into the other cell. But it's complete chance whether it's a paternal-maternal, maternal-paternal, and for example, you can get any combination of paternal and maternal, right? So that gives you, in all, about, I think there's probably about 8 million possible combinations for the type of egg cell that you get and the type of chromosomes in those cells. Now, the other important thing to know about autosomal DNA is recombination, or crossing over. Because not being content with just kind of shuffling up the chromosomes, before the chromosomes get shuffled, they cross over. So for example, here are 2 of my chromosome pairs. Here, let's say it's chromosome 6, and I got 1 from my father, 1 from my mother. Now, during recombination, they would cross over, and they'd be left with grandpa with a bit of grandma, or grandma with a bit of grandpa. So if my offspring would not have the same chromosomes that I have, because I would have recombined them before passing them on. And this is why you get segments, because the chromosomes are gradually and progressively segmented as they get passed down through the generations. So that is the basis of the genetic lottery, basically. Independent assortment shuffles up whole chromosomes, and you lose 50% of them each time they're passed on to a new generation. And recombination breaks up individual chromosomes, recombining them to form new chromosomes that are completely different from those of the parent, grandpa with a bit of grandma. And both processes start from scratch each time an egg or a sperm cell is formed. And each cell division produces a completely new, random assortment of DNA, and that is why you don't share the same DNA as your siblings. Now, I'm just conscious of time, and we probably have come to the end of as much as I can talk about, but what I will do is this will go up on the internet as a longer talk, So you've been very, very kind. I'm going to make way for Jared Corcoran. Is he here? Is Jared here? Yes. So I'm going to make way for Jared Corcoran, but the rest of the talk and the rest of the technique will be available on YouTube. Thank you very much for your kind attention.
Very